Okay, so thank you I want to thank Michael for inviting me to speak at this conference, which I really like. I'm a researcher in Marseille, in France, and my team specializes in work on membrane proteins and especially VDAC. So today I want to discuss with you how lipids can modulate the organization of VDAC in the membrane. So VDAC is the voltage-dependent anion channel, and it's often considered the metabolic door of the mitochondria because it's the most abundant protein of the outer mitochondrial membrane, and it's a main conduct for ions and metabolites. So ATP, that is most of the cellular ATP, is synthesized inside the mitochondria and utilized in the cytoplasm. So you really need to have this passage. And this is done by VDAC, that you can see here is a 19-strand beta barrel of 32 kilodalton, which has 3 isoforms in mammal, VDAC 1, 2, and 3. The 3 isoforms are able to exchange metabolites and ions. And this is originally how we discovered VDAC. And this was VDAC's main function for years. It was a metabolite exchanger. However, more and more, VDAC comes out as a major player in mitochondrial regulation. So not only in metabolite and ion exchange, but also in a lot of different functions. For example, in apoptosis, so cell death, where VDAC can interact with some of the pro-apoptotic or anti-apoptotic proteins and participate in mitochondrial outer membrane permeabilization, but also in lipid scrambling, calcium homeostasis with the ER, or mitochondrial DNA release. So we identified a lot of partners of different partners for VDAC. Some people claim that there is up to 100 putative partners for VDAC. And very little is known at the molecular level about VDAC roles in mitochondrial regulation. And this is kind of the topic we want to understand better. And so one can ask, okay, you have one protein, VDAC, and you have so many different functions, and how can one protein do all those things and sometimes be involved in antagonist pathways? So one of the way is that VDAC has 3 isoforms. So some isoforms are specifically implicated in some pathways. So for example, VDAC2 in apoptosis or lipid scrambling. VDAC1 is more in neurodegeneration, and we don't exactly know what VDAC2 is doing. Another way is through the different oligomeric states of the protein. And this is what I want to talk about today. Indeed, it was shown that VDAC oligomerization is key in mitochondrial physiology because it has been implicated in numerous physiological events such as lipid scrambling or apoptosis or mitochondrial DNA release. However, the determinants of the oligomerization, what is going to drive VDAC to assemble is very little known, even if we know that lipids have a role into this. And we know that lipids are important for membrane protein function and especially for VDAC. So we decided to set up and study what is the role of lipids in VDAC assembly. And for this, we are using atomic force microscopy, which is a very nice technique that allows a nanometer resolution. So it's very well suited to look at protein organization in membranes. What we do is we reconstitute our protein of interest, the isoform 1 of VDAC, into liposomes of controlled lipid contents. And then we absorb those liposomes on a mica surface, and then we can image it with the AFM microscope. And basically what the AFM is going to do is probe the surface. And because VDAC is a pore, as you can see here from the top, then you're going to have, this is a cross-section, the tip is going to go down the pore. And here on this topograph of AFM, I have larger image afterwards, but what you can see is every black little dot here is a pore. So it was already, so mitochondrial outer membrane were already imaged by AFM almost 20 years ago now. And so more than 50% of the protein of the mom are VDACs. So basically everything you could see here, every little dot, black dot is a VDAC, because in AFM, what is higher is in yellow and what is lower is darker. So basically all of these are VDAC pores. And already we could see different kind of organization with small patches here, very small patches of maybe dimer, monomers, hexamers. And basically we could already see that in the native membrane, VDAC had different organization. However, for us, what was missing here is what was the specific role of lipids, because you cannot control the lipid composition of those membranes. I mean, at the time they didn't. And we also had other proteins. So we decided to go ahead and look at one specific isoform, VDAC1, with the three main lipids of the outer mitochondrial membrane, PC, PE, and cholesterol. Okay, so this is a library. I'm going to go through every image after, but basically this is just to tell you that if you take one protein and you reconstitute it in the same condition, but just varying the lipid composition, then you can have a lot of different type of assemblies. Okay, so now I'm going to walk you through the first assembly that you're going to see here. Those are small patches of VDAC. And it really interested us because, as you can see, this is a large patch of VDAC. Every little black dot is a VDAC. And it's very stable in time. So, for example, in AFM, we image for several minutes. So it can be stable over like 10, 15, 20 minutes. And we call them honeycombs because at first it reminded us what of the structures the bee are forming. And it appeared organized to us. But then when we build a model using our molecular dynamic simulations data that I'm going to show you later, then this revealed really that this is not organized, but there is an intrinsic disorder. And what is very interesting to us is that in dark gray, you can see the VDAC molecules. Some are in direct contacts. But then in yellow is the annular lipids, the first layer of lipids, and in blue, purple is the other lipids that are not in direct contact with proteins. You can see that this patch is largely formed of lipids and is stabilized mainly by lipids-protein interactions. And so we have both tight protein-protein contact, but also lipid-mediated association between the protein. And so like the lipids are acting like kind of a mortar. And that was very interesting to us. The first thing we asked ourselves was, is it physiologically relevant? Because we are doing reconstitution, right? Okay, so we decided to first look at the orientation because VDAC was known to already reconstitute in antiparallel assemblies, for example, in the structure in bicelles. And so this is not physiological. And we cannot control VDAC orientation during reconstitution into liposomes. So we decided to check it. And for this, we used a method that we designed called nanobid mediated orientation determination, so we have a histag in our protein here in the IMS side, and we have a functionalized nanoparticle of gold. And so if the histag is visible, then the gold is going to fix. Okay, so now instead of having black dot for every VDAC, we now have a yellow dot, and this yellow means it's higher, and it's because a gold nanoparticle is going to on each of the pores and is blocking the pores. And so instead of having a well here, then we have a peak. And as you can see here, we can see them. Every dot here is a gold nanoparticle, and it covers every single VDAC in this patch, meaning that this patch is parallel orientation. And so for us, that was very interesting. And then we decided to compare what we got here with images from that were obtained in mitochondria, and you can see that it really resembles what we observe. And then we did some negative stain of outer mitochondrial vesicles, and you can see here also in negative stain every black dot here is a VDAC, and it really also resembles this kind of assembly that we see. So interestingly, using only 2 lipids of the mom in a ratio that would mimic what you have in the membrane, we can already reproduce native structures, and this was already something interesting. Then we decided to look at the effect of cholesterol, because cholesterol is the third major player in the mom, and it interacts with VDAC at different binding sites. So when you have 5% of cholesterol, we could see something that looked like a honeycomb, but smaller and maybe a bit more spaced. And you can see here, this is a patch that contains around 8 molecules. This is a small patch, and small patch is a bit larger, but that really reminded us what was seen in the patato-tuber mitochondria here, small patches. And then when you make a model when we made the model, then you can see really that even though those are very, very stable patches. You have very little direct contact between the protein. Some proteins are isolated, but still they're not diffusing. They're very stabilized by the lipid around them. And this is a rather loose packing, and that's often called glass-like. Then when you go to 7.5%, we see something that resembles more the honeycomb that we saw with that cholesterol. And so we have that really resembles what we see in the native membrane. And when you make the model, then you see that you have something a bit more compact than at 5%. And when we go to 10%, then we have, which is a high cholesterol content of the membrane, then the mica is covered with honeycomb up to 10 micron wide. But so you can see the areas that are kind of rough have the protein. And those areas here are actually just lipids devoid of protein. So we have patches containing only lipids, and it looks like the protein is going to take whatever lipids it needs and segregate the rest in those lipid lakes. And so you can see that here you have a very high compaction. And so now if I put side by side the different condition of cholesterol that we made, you can really see that the cholesterol increases VDAC local concentration and the compaction and size of the assemblies it can make. And to us, this is very interesting because you can have local variation of cholesterol in the mom, but also variation between the mitochondria. And I mean, cholesterol is something that can vary quite a lot in the mom. So one can imagine that different structures like this can coexist in the mitochondria. Then we tried different conditions. So these are only two examples, but basically we tried the same two main lipids that we got the first honeycomb with, but at different ratios. And what you can see here, we are losing the resolution because you have a lot more dynamics. We don't see honeycomb at all. It's more disorganized. Same thing when we remove PE. So we tried a lot of different conditions, and it really seemed that both the type of lipids and the ratios are important to see the type of organization that we see in the native membrane. Then we decided to rationalize what we saw using molecular dynamic simulations. So for this, we use coarse grain simulation. So we made triplicates with random starting points for lipids. And interestingly, after 20 microseconds of simulation, everything converged and gave something very reproducible. Basically, when we had a simple membrane of only PC and PE, you can see that VDAC sorts the lipids around it with PE preferentially in the first layer and PC in the second shell. Then when we add cholesterol, then it's even more dramatic because cholesterol has a higher affinity for VDAC and is really tightly bound to the protein, which we already saw before in previous studies. And cholesterol has a tendency to displace a bit of the PE molecule. So VDAC likes cholesterol, then PE, and has a bit less affinity for PC. And it's interesting because we just showed that PE and cholesterol are important to form honeycombs. And so this is important because it came back to other studies published in our group, and that shows that protein, and in this case VDAC, can selectively recruit lipids to form a lipid annulus. And this annulus can modulate the protein-protein interactions and the assemblies the protein can make. So just to conclude, I think, so this is just a schematic representation of the different conditions we have with no cholesterol and increasing amount of cholesterol. Then you can see really that talking about oligomerization can be misleading. I'm just saying that because a lot of people talk about, especially in the VDAC field, of VDAC oligomerization. So when we say VDAC oligomerization is important for this, it can be a bit misleading because even though we have some defined oligomer structures for VDAC, so we have some specific dimers defined, and an hexamer was published for the yeast for one. In what we are looking at here in membranes, it can make more sense to talk about clusters than really oligomerization that is not really well defined. And so to go back to the physiology, I think this is important because, as I was telling you, VDAC has a lot of different functions. and those different functions coexist in the mitochondria. And so, for example, a single channel can transport ions and metabolites, whereas lipid scrambling can only be done if you have a dimer or higher order oligomer. In sperm mitochondria, it was shown that a VDAC array is necessary to dock some of the protein of the cytoskeleton. And then it's important also for calcium flow. I'll just go back to that after. But if different lipid environments favor different organization, that means that very quickly, by changing the lipid content of the membrane, you can change the organization of the protein and you have another level of regulation of the function. And also, as I told you before, MOM is not homogeneous, and you can have different assembly depending on different lipid ratio, some contact sites. And so this is my last slide. So just to give an example, at the mitochondrial ER contact site, this is where the cholesterol gets imported, most of the lipid gets imported from the ER. And you have a high calcium flow through VDAC. And this is a very important thing, this calcium flow, because it's a passive diffusion, but you want it to be very efficient, because if calcium goes in the cytosol, it can be toxic. And often this is represented like this. But if you look at IP3R here and VDAC, one can understand that it's not going to be a one-to-one stoichiometry. And it was also shown in 2019 that IP3R forms large cluster. So if you have a cluster of IP3R, if this is viewed from the side and this is viewed from the bottom, then you definitely need a cluster of VDAC. And these honeycombs could be the cluster of VDAC that will ensure a fast calcium diffusion to the mitochondria. Okay, so this work was performed in Marseille between our team at the CNRS and the team of Ignacio Casuzo in CERN and at the University of Aix-Marseille. And thank you for your attention.
Okay, wonderful.